Hello everyone. In this video, we are going to discuss how to investigate the effect of varying temperature on the rate of photosynthesis. That means if there is any change in the temperature, like temperature becomes lesser than the optimum temperature, or if temperature increases beyond optimum temperature of the enzymes, so how it affects the rate of photosynthesis in any plant. Now, in order to investigate this, you have to arrange the entire experimental setup like this. Like it's shown here in this picture. Okay. So you require a beaker. First, you have to take a beaker and fill water in it. Fine. And then you have to take any aquatic plant. Okay. Now, this aquatic plant could be hydrilla, or it could be Canadian pondweed or Elodia, anything. Okay. You have to take all these aquatic plants all together and insert it in the stem of the funnel. Okay, so this is called stem of the funnel. Okay, so you have to insert all the cut ends of the plant into the stem of the funnel. Okay, and then you have to place this funnel into beaker full of water in inverted position, in upside down position. Then you have to take a test tube as it's shown here in the picture. Okay, this portion is called brim of test tube. Okay, the top part of the test tube is known as brim of the test tube. So you have to fill water till brim of the test tube, and then you have to place it on the stem of the funnel in inverted position as it's shown here in the picture. Okay, next you have to use thermometer in order to just to check the temperature of the water, whether throughout the experiment or throughout taking the reading, the temperature of the water is maintained constant or not. Okay, and in order to perform photosynthesis, you require a continuous supply of light. So we are using here a table lamp as a source of light. Okay, and this table lamp is actually placed here at constant distance. Now remember this thing very well, that whenever we perform any experiment, we have three types of variable in that. One is independent variable, okay, independent variable, then dependent variable, and last one is controlled or constant variable. Okay, so independent variable is generally only one factor, or only one variable is considered as independent variable. Now here we are actually investigating the effect of temperature on the rate of photosynthesis. So if we'll look carefully, what is actually controlling the entire process of photosynthesis? The entire process of photosynthesis is actually controlled by the temperature here. Okay, so this temperature here becomes your independent variable, because the entire rate of photosynthesis or entire rate of reaction is dependent on temperature. Here we have used a hot plate. Okay, this is a hot plate. The entire experimental setup has been placed on hot plate, because this hot plate provides the constant temperature to the water in the beaker. Okay, and this temperature will be keep on changing, or keep on changing in the sense, first we will perform this experiment on 25 degrees Celsius, then maybe on 30, 35, 40, 45, 50, like that. And if we want, we can go below 25 also. Okay, it can be on 20, 15, 10 degrees Celsius. Okay, so we can start from 10, 15, 20, 25, 30, 35, 40, 45, 50, like that. Okay, so this hot plate is used here as a source of heat or as a source of temperature. Okay, and this temperature is acting here as independent variable. So in this experiment, independent variable is temperature, and the unit of temperature here is degrees Celsius. So whenever you label this dependent and independent variable on graph, so always remember you have to label it along with the unit. If you don't mention unit, then you won't be awarded 1 mark for labeling of the axis. Okay, when we talk about dependent variable, so due to the change in temperature here, what is getting affected in this experiment? The rate of reaction, or you can say, the production of oxygen bubbles, or the rate of photosynthesis is getting affected. So rate of photosynthesis, or the rate of oxygen production or rate of reaction becomes your dependent variable. Okay, so independent variable is temperature and dependent variable is rate of photosynthesis, and other factors which are supposed to be kept constant or controlled throughout this experiment is the distance of the light source and then the intensity of light. Okay, this is very important. Intensity of light, distance of the light source, then the type of light given to the plant. Okay, this is very important, type of light, because plants, they perform photosynthesis on one particular wavelength. Okay, that is 680 nanometer to 700 nanometers. Okay, some plants use 680 nanometer and some plants uses 700 nanometer. Okay, so the similar type of light has to be provided at every temperature or at each temperature. Okay, and then this photosynthesizing aquatic plant is also controlled variable. That means when you are taking reading at 25 degrees Celsius, so the plant which you have used here or the type of plant which you have used here, same type of plant or similar number of plants you have to use while taking reading at 30, 35, 40, 45, 50 degrees Celsius also. Okay, you cannot change that. So this is your control variable. Apart from this, another very important raw material for the process of photosynthesis is carbon dioxide. Now here the water which we use is generally tap water, and tap water is having very less amount of carbon dioxide in it. Okay, and almost nil. Fine. So what we do, we add here a salt which is called sodium hydrogen carbonate. We can also use potassium hydrogen carbonate. So whenever these salts are dissolved in water, they liberate carbon dioxide gas. Okay, and this carbon dioxide gas gets mixed with water and it is used by plants in order to perform the process of photosynthesis. So the amount of hydrogen carbonate added in the water, okay, that means the concentration of sodium hydrogen carbonate solution should be same at every temperature. Okay, so this is also your constant or controlled variable. Okay, so this is how you have to set up the experiment, and then you have to take reading. Okay, then you can start with 10. At 10 degrees Celsius, in first 1 minute, how many oxygen bubbles were produced. Okay, how to take the reading, you actually have to count the oxygen bubbles produced here. Okay, you can easily notice the production of oxygen bubble after placing the entire setup in bright sunlight. Okay, so you have to take 2 readings at every temperature. Okay, and then you can just find out the average of readings, average of 2 readings at every temperature to get the mean value. Okay, so like this you can just record your readings, and then you can just plot a graph as it's shown here in this picture. Okay, so this horizontal axis is actually your x-axis. Okay, this vertical axis is y-axis. On x-axis we always label independent variable. Okay, and on y-axis we always label dependent variable. Fine. And independent variable in this experiment is temperature. The unit of temperature is degrees Celsius, and independent variable here is dependent variable here is rate of photosynthesis. Okay, so that is being labeled on y-axis. Now as you see this graph, slowly as the temperature is increasing, the rate of photosynthesis is also increasing. Okay, you can make out here the rate of photosynthesis is also increasing. Okay, so the first very important point is, as temperature increases slowly, the rate of photosynthesis or the rate of reaction also increases. Why? Because at lower temperature actually enzymes are there. So these enzymes are involved in the process of photosynthesis. Okay, so at lower temperature what happens is the kinetic energy of molecules, or the raw materials or the factors which are involved in the process of photosynthesis. So at lower temperature what happens is the kinetic energy of molecules is very low. Okay, so as we increase temperature slowly, the kinetic energy of these molecules increases. Fine, the kinetic energy of molecules increases, and hence the collision between the substrate and enzyme increases, and the rate of reaction increases. And as temperature increases, the rate of reaction also increases up to a certain point. Okay, and that point is called optimum temperature. Okay, so optimum temperature is the temperature at which plants perform photosynthesis at the fastest rate. Okay, the rate of photosynthesis is highest at optimum temperature, and that temperature in plants is generally between 20 to 25 degrees Celsius. Okay, so most of the plants, they have this optimum temperature of 20 to 25 degrees Celsius. Okay, so in plants, enzymes, they work best in any of the reaction. Enzymes, they work best at optimum temperature. But what happens when temperature goes beyond optimum temperature, like 30, 35, 40, 45 degrees Celsius? Okay, so as temperature increases beyond optimum temperature, you can notice here, as it's shown here in the graph, the rate of reaction or the rate of photosynthesis decreases completely. Okay, it goes down. Now the reason for this is, I've told you that in the process of photosynthesis, enzymes are involved. Now the reason for this is, in unit 5, that is enzymes, we have studied that if temperature goes beyond optimum temperature, the enzyme gets denatured. Okay, so this point is very important. Enzyme gets denatured. Now the meaning of this term is, every enzyme has its active site. Okay, active site is a place on enzyme where any substrate molecule comes and gets fit into the enzyme. Okay, for example, this is an enzyme and these points are the active site of the enzyme. This is a substrate molecule. So substrate molecule comes and gets fit into the active site of the enzyme. Okay, and then they form enzyme substrate complex. Okay, so what happens is, as the temperature goes beyond optimum temperature, the enzymes, they get denatured. That means the shape of active site of the enzyme changes. Hence what will happen, as the shape of active site of the enzyme changes, so the substrate molecules cannot fit into the active site of the enzymes, and finally the rate of photosynthesis will decrease. If you'll remember, when we have discussed about the effect of varying concentration of carbon dioxide, or the effect of varying light intensity on rate of photosynthesis, after certain point the rate of photosynthesis was getting level off, was becoming saturated. It was becoming constant. There, the reason for becoming constant was any other factor, could be limiting factor. Okay, but here the rate is completely decreasing, and finally the process of photosynthesis stops, because at high temperature all the enzymes, they get denatured. So this point is important. All these 3 sections, you can take it as 3 sections. 1, okay, then this is 2 and 3. So you have to remember these points very well. In section 1, as temperature increases, the rate of photosynthesis increases due to the increase in kinetic energy of enzyme and substrate molecules. Due to increase in kinetic energy, what happens? The frequency of collision increases. Okay, hence enzyme substrate complex gets formed very easily or faster. Fine. In second, you have to remember the rate of photosynthesis is highest at optimum temperature, because that is the temperature at which enzymes, they work best. Okay, and after optimum temperature, what happens? Due to increase in the temperature, enzymes, they get denatured. Now you should know the meaning of denatured. Denatured means the shape of active site of enzyme changes. Hence the rate of photosynthesis decreases. Okay, so this was all about the effect of temperature, or effect of varying temperature on the rate of photosynthesis. In next video we will discuss about limiting factors. Take care till then. Bye bye.